Hey guys, what's going on? So today we're going to be starting a new playlist in biophysics. Typically you won't find an all-encompassing course on YouTube on biophysics. This is what I intend to do for this playlist. It's going to be long, it's going to be relevant, it's going to be interesting, and it's going to be fun. Right, so what I intend to do here is cover from the molecular, all the protein, DNA stuff, to organelle-sized physics. And when we do that, we are going to be talking about thermodynamics, we're going to be talking quantum mechanics, we're going to be talking optics, we're going to be talking a lot of areas in physics that are used to study areas in biology. Namely, this is why it's called biophysics, right? We're going to be talking about cryo-EM, we're going to be talking about SPR, we're going to be talking about different types of methods that help us understand the biological world around us, to help you get a job in wherever you want to go, be it pharmacy, be it academia, be it research. This is sort of going to be the all-encompassing biophysics course to go to if you're interested in this kind of stuff. Again, if you like this kind of content, make sure to hit that like and subscribe button, and also make sure to go into my Patreon page if you want to support this channel, where I will be creating summary sheets on this playlist for you guys to keep for yourselves. Now let's get into the topic. Okay, so we're talking about above and beyond biophysics. That's the name of this playlist, and today we're going to be talking about DNA structure and function. What is DNA? DNA, simply put, is a bunch of sugars attached to each other via phosphate links, right? So these sugar backbones, it's a total of 5 carbons, right? So we start counting here. We start counting right here. This is carbon 1, carbon 2, carbon 3, 4, and 5. We can do the same thing here. 1, 2, 3, 4, and 5. And 5 and 3 are linked to each other via this phosphate group. This total thing is called a nucleotide. Okay, a nucleotide is one of these things, these 5 carbon things, or sugars, if you want to call it. You can call them things, where we typically call them sugars in biochemistry. But if there's a phosphate involved, it's considered a nucleotide. If there's no phosphate involved, it's considered a nucleoside. DNA is a nucleotide, and the bases that make up the DNA consist of one of 2 different types of bases. Now let's take a look at what these bases look like. In one case we have purines, right? So this is going to be adenosine and guanine. So A and G is how we're going to abbreviate that. Those. And then we have pyrimidines. Pyrimidines, we have threonine, cytosine, and then this one here is going to be uracil. Uracil, I'll say that in brown. It's going to be on RNA only. We're not going to talk about RNA yet. We'll talk about that later. Right, so the 4 key players when it comes to DNA are going to be A, G, C, and T. Adenosine, guanine, threonine, and cytosine. Okay. So this is how they look like when we actually look at, if you actually look at a structure of DNA in a textbook or online, the A and the T are always going to match up with each other, right? So here is our A, and here's our T, right? So we have our A here and our T here. They attach sort of at an angle of with respect to this sugar. This also attaches at an angle with respect to the sugar. Notice here how these sugars are upside down, right? This upside down nature is called the 5 to 3 prime. So if we're reading in this direction, right? If we're reading in this direction, actually no, wait. Let's read in this direction. If we read in this direction, we're going 3 to 5 prime, and in this direction, we're going 5 to 3 prime. Okay, so we have anti-parallel strands, if you will. We have anti-parallel strands. Each strand is attached to a sugar, right? Or is attached to a base. So each one of these sugars attached to a phosphate is attached to a base, right? And these bases are hydrogen bonding to our adenosine or to our other bases, right? So here I'll write A, and then my G is in, looks like that's a magenta color. This is G. And then here is our C, and here is our T, which I have in blue. So that's DNA. In RNA, things are going to be a little bit different, right? In RNA, we're going to have something right here. We're going to have something right here. Well, that's what we have right there is going to be an OH, but that's for RNA. We're not going to talk about that right now. We're talking strictly DNA, right? Now I want to talk a little bit about combinatorics. What I intend to do for this playlist is make this a little bit different than what you typically see on other videos regarding biochemistry, biophysics, chemistry, what have you. And what I want to do is I want to take a look. Okay, so we've taken a look here, and we can ask a couple of questions about this DNA, because the essence here of DNA is that it's stored the sequence of nucleotides. Say we're reading in the 3 to 5 prime direction. We have T, A, and then suppose we have G and C after those. And the question we want to ask now is, suppose we have four bases. What are all the possible number of permutations that we can have of those bases, right? And that gives us a sense of how much information could theoretically be stored in DNA. So for a tetranucleotide, that's 4 bases, the number of possible combinations in the order, if the order is random, is going to be 24, okay? And here's why. So we have A. We're going to start all these sequences here in A, right? So 2 of them are going to be T, the 2 of them have the next letter being T, the next 2 have the next letter being G, and the next 2 have the next letter being C, and then we can permute the last 2. We can sort of do an anti-permutation of the last 2, okay? And so, and the idea here is once you pick the first one, okay, you've used that up. So you have 4 possibilities in picking the first one. You have 3 possibilities in picking the second one because you've already picked the first one. Then you only have 2 possibilities in picking the third one, and you have only 1 possibility in picking the last one. And so when we multiply all this together, that gives us 4 factorial, or 24, right? So we take a look here. 4 times 3 is 12. 4 times 3 is 12. Times 2 is 24. Times 1 is 24. So these are all the possible permutations we can have if we only are looking at 4 different nucleotides. Okay? We also want to keep in mind here that A and T, the number of hydrogen bonds in each, right? So between A and T, we have 2. Between T and G, or not T, right? So this is going to be, this here is T, right? That was, and then this here is C. All right, my bad. So we have more hydrogen bonds here. That means the degree of interaction is going to be stronger between G and C. Okay, and that's something to keep in mind also. For an N nucleotide, the number of possible combinations is going to be N factorial. Okay, so suppose we had 10 nucleotides. So if we have 10 nucleotides, it's going to be 10 factorial. That's going to be a big number. Okay, so the more nucleotides we have, the more number of permutations we could have, and that is that's a measure of how much information could theoretically be stored in one strand of DNA. Now keep in mind that DNA is hundreds of thousands of bases, so 100 or 1,000 factorial is going to be a really big number. And that's why we get so much degree of variability in the outcomes of DNA transcription and translation. Okay, and we're going to see how biology sort of trims that down so all that variability can at least get somewhat compressed into to not such high degrees of information. We'll see that later. So suppose now we have 4 empty boxes. So what I want to do now is we want to sort of limit. So this here was was us taking a look at if we had a certain number of bases. This was sort of the base view of the combinatorial question on DNA. Now suppose we had nucleosides, right? So we have, say, 4 nucleosides, and we have an infinite number of bases at our disposal. Okay? And if we have an infinite number of bases at our disposal, we want to ask the question now, if we have what I'm going to call 4 empty boxes, these are going to be the nucleosides, and we could fill each box with one of the 4 nucleotides, or one of the 4 bases. I'll just call them nucleotides for right now. Well, in one case, say we have G, G, T, and A. Okay, this is one possible situation we can have. Another situation with we can have G, G, T, and G. Right. This can go on and on and on. If we had an infinite number of bases at our disposal, but we only had 4 nucleosides, then each one of these is going to have a 1-4 possibility of being A, G, T, or C. And so if each one of these is 1 and 4, then this changes, right? So 4 times 4 times 4 times 4 instead of 4 times 3 times 2 times 1. And in that case, we get 4 to the 4th power, and this is going to be 256. Right? So if we have a limited number of bases, the number of possibilities is only 24. Whereas if we had tons and tons and tons of bases at our disposal, but only 4 nucleotides could be occupied, then the number of possibilities was way, way bigger. Okay, and this is another combinatorial problem that DNA in biology is going to have to contend with. This idea that you could easily make a lot of bases. You could have hundreds and thousands of bases at your disposal, but you can only fill 1, 2, 3, 4, maybe 100 nucleosides. Well, this discrepancy between 24 and 256 that's a big discrepancy, and this again alludes to high degrees of variability within the potential outcomes that DNA has to offer when we start talking about how it transcribes proteins and all this kind of stuff. So let's now constrain this a little bit, okay? If we had n nucleosides, then the number of possibilities is 4 to the n, right? Because each one of these is 4 possibilities, so that's the base. So n is 4, okay? In this case, because we have 4 boxes. So we can also, if we start imposing some constraints here, suppose we have these bases, right? Some of them are going to be, and some of them are going to be radioactively labeled. You can do this kind of thing. You could say, okay, suppose we had 2 adenosines, 3 cytosines, and 1 guanine, and we radio label 2 of these guys and this guy. Notice how 2 of the radio labels here are different. If we do this radio labeling, then order is going to matter when they get applied or added onto the DNA strand of nucleosides. And we have, so the potential nucleotides, right? I'm going to call these nucleotides because we're going to assume there's a phosphate there. Nucleotides. If we ask the question, okay, we have 3 nucleosides, and or we have 3 boxes, right? So we ask the question, we have 3 boxes, what's the degree of permutation going to be if we have only 6 spaces at our disposal, but order is going to matter, right? Order matters, meaning we've radio labeled these things and we can theoretically look at where they are with respect to each other on the chain. Then we can say, well, here's a potential outcome, here's a potential outcome, and here's another one, and this could go forever, right? Not forever. We have actually a limited number of permutations we can have, because we're only working with 3 boxes, and these things are radio labeled, so order is going to matter. Well, in that case, this is going to be a combinatorial question, and this question is answered by a permutation. Okay, if we get given some 3 nucleosides, empty nucleosides, and we have some number of bases, what are the possible number of permutations that we could have, given that these guys are radio labeled, or given that we can actually determine what the order is with respect to each other, and that this is going to be this equation, right? So we have our factorial, and that's going to be divided by the factorial of the difference between the number of bases and nucleotides. Okay, so this is a permutation, right? Let's take a look at what combination addresses. So combinations address the question of, okay, order is not going to matter, right? So if you think about it, if order doesn't matter, then redundancy is going to happen, okay? Because you can have this situation, right? This time things are not radio labeled, okay? This time we're saying we don't think we're going to be able to discern the order of the outcome with respect to each other. And so I'll erase this for right now. These are just, we'll call these outcomes, right? I'll just call these outcomes for right now. And so we could have this potential situation where this accounts for, say, this potential situation accounts for something that looks like this, right? Where it doesn't matter if things are radio labeled. Those 2 outcomes are the same because we don't have them radio labeled, right? And so this number is actually going to have to be smaller than the number we get in this case, because we're going to have to deal with that redundancy. So this here is in other words, this here is 2 states in this case, okay? And so we have to divide by the number of nucleosides, and there's a factorial right there. So this is what combinations address, right? So combinations address this question of really ordering. The order of sequences doesn't matter. Okay, the order of bases doesn't matter, and this is going to be something worth keeping in mind later, because my whole thing about this biophysics approach is going to be how can we trim down the degrees of information that in going from DNA to protein, okay? Because we have so much, thousands and thousands of degrees of variability in DNA, in this DNA space, and we want to trim this down so that we can understand how proteins emerge and how things get modified, so that we can get a finite number of proteins that will have some sort of finite function or functionality to them. And so these are the 2 equations that we've come up with, right? So B is bases, N is the nucleosides. Okay, so given some number of nucleosides and given some number of bases, what are all the degrees of permutations and combinations that we can have? That's given in these 2 equations. All right, notice how PBN is equal to, or it's going to be 1 over n factorial is going to be equal to CBN. Right, so this is kind of interesting to look at, and we want to keep that in mind as well. All right, so suppose now we have 3 nucleosides, codons, and an infinite number of bases. How many possible combinations do we have? So I'm calling these codons for a reason, because codons are the basis of building proteins. Well, we've addressed this before. 4 times 4 times 4, right? Because we have 3 possible nucleosides, and so what are all the potential random numbers of permutations can we have in this case? Well, that's going to be 64, right? So 4 times 4, 16 times 4 is 64. And this is the basis of the codon chart. So if we look at this chart, we have 4 times 4 times 4. Okay, 64 different possible outcomes where U here is a replacement for T, right? Because when we're talking proteins, building proteins, we're moving away from threonine, which exists in DNA, and we're going to RNA, which we're going to talk more in detail later, which consists of uracil instead of threonine. But anyways, this 64, we think of this, the 64 is the basis of this table, and you'll see this table everywhere. And notice a few things, right? So leucine, serine, and arginine. If you look at this table long enough, we have leucine, serine, and arginine, and arginine here and arginine here. These 3 here have the highest probability of occurring, given if all of this was random, right? Because we have 4 plus 2 is 6. Serine, 2 plus, where'd the other serine go? 4 is 6. Arginine, 4 plus 2 is 6. Everything else occupies either 4 triplets or 2, right? Leucine, serine, and arginine have the highest probability of occurring, which is kind of interesting. And we're going to look at this a little bit more as well. Leucine is hydrophobic, serine is uncharged, arginine is positively charged, given physiological conditions. We're going to go into this a little bit deeper. I'm sort of priming us for what's to come. And last but not least, I really want to hit home 2 big pictures here when we talk biophysics. One picture is going to be the central dogma of biology. That dogma is that DNA gets transcribed into RNA, which gets translated into protein. We are essentially going from nucleic acid, nucleic acids being converted into amino acids. So this conversion takes us from information space, right? DNA consists of all the information, to function space, where proteins do all the functionality. So there's this mapping between information space and function space when we're talking biology. Okay? And these arrows, again, transcription is going to be done by RNA polymerase plus some other proteins, but RNA polymerase is the big player. Translation is going to be done by the ribosome. The ribosome is the big player in taking RNA and converting it into protein. This is where those codons play a big role, right? Because codons are triplets in the nucleic acid space that get converted into amino acids. Okay? And then we have this replication, which is done by helicase and DNA polymerase. And we're going to talk about those also. So that's the central dogma of biology. The other thing we want to look at really quick is the mathematical structure of biology. What does biology look like from the perspective of, say, a physicist or a mathematician? Specifically a mathematician, because mathematicians really love maps. Right? They really like the idea of going from one space to another space, and what's the mapping function that does that? Well, in biology, there's actual biological processes that take us from sequence space to function space. Now let's take a look at some of these, right? So we go in from sequence space, which is DNA, this is information space, to function space. This is where proteins live. We go nucleic acids to amino acids. And what's very, very interesting is at one point in sequence space, say one DNA sequence, can actually get blown up into different points in function space. And the way this happens is through multiple mechanisms. One mechanism is through splicing, right? So you transcribe some nucleic acid, and then you take chunks of it out, right? And you glue the rest together. That's splicing. Okay. And you could, depending on which chunks you take out, you create a different type of protein, right? Creating homomers or heteromers, right? So given one point in sequence space, or 2 points in sequence space, you can actually take that sequence, copy it, or slightly modify it a little bit, and take that copied version and then attach it to the other one, right? And that gives the resulting protein some sort of functionality as well. We can do histone binding, right? So histone binding is this very interesting way of different types of proteins attaching themselves onto the DNA and modifying what gets transcribed and what doesn't, right? So the histone binding takes us to some different function space as well. Or some different point in function space as well. And then last but not least, post-translational modifications. So depending on the environment, what's around at the time, the DNA sequence that gets transcribed can get post-translationally modified, and then the protein gets produced, and this protein has some sort of different type of function as opposed to if it was post-transcriptionally modified in a different way. Okay, this is the mathematical structure of biology. So we've talked about combinatorics, we've talked about the central dogma of biology, we've talked about DNA, right? So this is the foundation. In the next video, we're going to be talking about amino acids, their properties, how they make up proteins, and we're going to go from there. Again, if you like this kind of content, make sure to hit that like and subscribe button, and I'll see you guys in the next one. Bye.